My name is Agata Kiełkiewicz, and I represent the clinical trial sites network, which collects the data mentioned in the previous presentation. As already said here a million times, the market is growing very, very fast, so that means that the number of clinical trials is growing as well. I would like to show you a slide which was used by my colleague who's here with me today in 2018, and she presented the data that the number of recruiting studies at June 2018 was 146. To compare, today we have over 790 studies, so it's over 5 times more. And also, over 3,276 studies is already registered in the clinicaltrials.gov database, which is official database of the trials conducted all over the world. I just put some numbers to show you how many of specific studies are here. Like in smoking cessation, we have already registered over 1,000 of studies. What can be done? As was mentioned before, the data collected from the clinical trial, we can run the pharmacokinetics assessment, we can assess the biomarkers, we can assess the safety profile or even the effectiveness, or even the adverse events connected with intaking the nicotine or nicotine-containing products or any interactions. I divided into 2 points, which the first one I've just said is the physical one, and the second one is the psychological one, because we also can check the nicotine craving, we can check the satisfaction from use, we can check the relief of withdrawal of symptoms, product liking, etc., etc., etc. And I think much, much more. Maybe even I don't know some of them because this market is growing so fast that I might be not familiar with all of the things we can choose to be assessed during the clinical trial study. What's important to have the highest possible quality of data? One of the things is to choose the right site which will be able to perform that kind of studies, especially in the healthy volunteers, which are very hard to conduct, being honest. What more is the access to the smoking population, which is obvious, and qualified and experienced team members. I'll show you in a moment why. The quality and the good recruitment plan is the basis of a satisfying clinical trial. And here, I want to show you the main thing, I would say, of the study, because this is the start, the planning, the beginning. Because in this kind of studies, there are so many procedures in sometimes even a couple of minutes, like before intake, 3 minutes after intake, 5 minutes after intake, and these are the procedures which are a number of, and you have to remember that usually at the same time we have a number of participants, like 10 or even 20. So the most important thing in the beginning is a planning. A very, very careful planning is taking the protocol, having the procedures, and analyzing one after one after one if it's doable if it's possible, how many team members we will need to conduct this procedure at the time to meet all of the criteria. That's why we have the quality controls, we have staff training, we do the mock trainings before the study starts. This planning is very, very careful and with very, very close cooperation with the sponsor. All of the documentation is prepared already. All of the procedures are prepared already. We have this huge worksheet. When we put all of the volunteers, we put all of the procedures one after one, minute after minute, and we can cross-check then if we can do it, if it's doable, if some of the procedures will not fall down the whole study or the whole visit. That's why we also analyze the holidays, Christmas, etc., to be able to have these volunteers on site, wanted to participate in the study, to meet the recruitment plan, and to meet the retention. So, as I mentioned, the recruitment strategy is one of the most important issues. So this has to be planned as well. We do have the call center, very experienced team who contact the participant, the whole department, the SOPs procedures, and just to show you, to randomize 100 people into the trial, we have to invite for the screening over 400. And that means, because not everybody will agree to come, that means that we have to contact over 800 people. We have huge database, so we can do it. It's doable for us. But I just want to show you the numbers, the difference, how much people has to be conducted to meet the criteria of the protocol and be able to be randomized into the study. According to retention strategy, in the terms of the long-term studies, and in long-term study, I understand the study who can be conducted for 7 months. So the participant has to come for the visit for the 7 months. It's a long time, actually. So in this case, our retention level is around 95% or more. In a term of the short studies, this is probably 100%. We have a very good contact with our participants. They are called, they receive the SMSs about every visit, the reminders. So they are quite connected with us, they trust us, and they want to come to the visits. They want to participate in the study, and when they finish the study, they usually asking us for more, if we will have something, some study that they could participate. These pictures shows how this really looks. Those who were here last year might remember this picture from Teresa Brodniewicz presentation, who were here last year. But this is how the study looks like. In here, we have just 2 volunteers and 6 people who are team members. In this, let's say, up to 10 minutes, they are smoking. They have the blood draw. They have the vital signs. They have the questionnaires to be completed. So please imagine how this has to be planned. To avoid any mistake, and it has to be, if the blood collection is at minute 3 after, let's say, smoking, it has to be at minute 3, not at 4, not at 2. This is our experience, but what I would like to say, imagine, please, how to plan the study for 320 volunteers. And they were 7 days on site. Thank you very much. I'm here with Marta Zakrzewska, as I mentioned before, to discuss all of the questions, all of the opportunities that can be.
Thank you very, very much.
Thank you.
Thank you very, very much, Agata. Vitally, vitally important stuff. Questions? Questions? Over there, over there.
Thank you for that presentation, Dr. Marewa Glover, New Zealand. Just wondering what the payment is, because that's a very impressive retention rate.
Well, they are paid. Well, it all depends on the scheme of the study and the number of the visits.
Ian.
Sorry, I just wanted to kind of pick up on Mariah's question. It isn't necessarily a payment, Mariah. I've conducted studies at MTZ. They do exceptional research, and we've done PMTA studies with them. It isn't payment as such. It's, I guess you would call it IRB-approved compensation for the time. And as Agata said, if they're undergoing multiple procedures, blood draws, nights in the clinic, etc., they're compensated for that time to a degree which the ethics committee has approved.
That's correct, I confirm.
Yeah. One more over there, and if you don't mind, I think I better make this the last, because we are a little bit behind.
Thank you. Brilliant. Thank you very much for the presentation. Ben Kelsey, I work for a design consultancy in Cambridge, UK. I was interested on, I think your third chart, you had a list of things that you measure, which I assume are wet tests, and there are some psychological factors as well. I was just wondering what your means for data collection for those things would be.
Again, please.
So you mentioned that you collect psychological factors or psychological data. How do you collect that data from your participants?
By the questionnaires. We collect the questionnaires in timing as well as the pharmacokinetics.
I have one other quick question as well, if there's time. Do you, all the biomarkers that you collect, are they wet, through blood samples and saliva samples, or any of them digital biomarkers through smartwatches or other means for testing?
I would like to ask for a repeat.
Maybe Ben, follow up on that outside.
I would.
That would be better. Well, thank you very, very much, Agata. That was great, and thank you.
Thank you very much.